Hello and welcome back. So what I want to do now is just focus more on these defensive functions of some of the compounds I've already mentioned and some new ones that I'm going to talk about in this video. So many of these secondary metabolites that affect insects do so by disrupting their physiological processes and metabolism. And yet, we actually really don't know a lot about how that exactly happens for a lot of different systems. But what we do know is that they appear to disrupt membrane function, so they can influence and inhibit nutrient and ion transport. We also know that they can deter signal transduction processes. That can alter metabolism as well as hormone controlled processes. We also know that some of these compounds are structurally similar to neurotransmitters and have been shown to interfere with insect neuroreceptors. And what I want to kind of end with here when we're talking about this is that all of these responses and this toxicity in general is incredibly dosage dependent and depends on the dosage over time. So that is really a function of the insect's age, its size, its health, and the mechanisms that that specific species uses to either absorb or excrete secondary metabolites. And then finally, I just want to kind of talk about the plant for a second, because some of you might be wondering if plants are making all of these toxic compounds, how do they avoid autotoxicity? And the answer is that many plants have evolved these specialized storage structures that minimize self-toxification. So we're going to talk about a few examples where they're stored in the vacuole. There's also examples where they can be stored in the apoplasm. And in conifer species, like pines and spruce and firs, they actually are stored in structures known as resin ducts. Okay, so in my first video, I mentioned that there are three major classes of secondary metabolites, and here I'm just going to go into a little bit more detail. And first start by telling you that the three classes are down here at the bottom, the first of which is nitrogen-containing compounds. Then we have phenolics here and terpenes. And what I want you to notice from the schematic is that all three of those groups, compounds within all three of these groups, are derived in some way from products of primary metabolism, which you can see here. So at the top, you see CO2 and water entering the plant. We see the plant undergoing photosynthesis. And as a result, we have a lot of photosynthates and primary metabolites that are here. And these actually are serving as those substrate molecules that enter these various biosynthetic pathways and create the different natural products. So what I want to do in this first video, though, is just focus on one group, which is the nitrogen-containing group of secondary metabolites. So nitrogen-containing secondary compounds are encountered less commonly in plants than phenolics and terpenoids, but as you will soon see, are incredibly important when considering plant-insect interactions. So the thing about these compounds is that they're synthesized from amino acids, both aliphatic, which are just linear chain amino acids from the TCA cycle, as well as aromatics, which are these unsaturated ring compounds that are derived from the shikimic acid pathway. And just in case you forgot about amino acids, I'm just putting up an amino acid structure here to give you a visual. So amino acids, if you'll recall, have this central carbon molecule or element. And then attached to that is an amino group, a hydrogen, a carboxyl group, and then the R group. And it's really this R group that makes the amino acid very unique, where it can cause the protein to be more hydrophobic or hydrophilic or more acidic or more basic, et cetera. So again, with amino acids, it's really that structure dictates function for proteins. And that's going to be important when we get to the non-protein amino acids that I'm going to talk about today. Okay, so within this group of secondary compounds known as nitrogen-containing compounds, there are kind of these, four classes within it. And those are alkaloids, cyanogenic glycosides, glucosinolates, and non-protein amino acids. And so I'm going to go through each one and just kind of give you some cool examples of their function and how they impact herbivores and humans, actually. Okay, so the first group are known as alkaloids. So alkaloids are present in about 20% of higher plant families, including the ones that I'm showing you here. And one that might pop out to you might be the Solanaceae family, which we talked about in the first video with deadly nightshade. But in that family, it also includes tomatoes and eggplants and chili peppers and plants like that. So above, I'm showing you some structures for some common alkaloids: caffeine, theophylline, and theobromine. And so if you are a lover of chocolate and tea, like I am, then you are definitely ingesting theobromine and caffeine. And theophylline is also found in tea, but not typically found in chocolate. But what you will know about all of these is that they have stimulating effects. And that is the same for insects. So overall, alkaloids are a really structurally diverse group of compounds. There are more than 12,000 that have been identified to date. And what makes them true alkaloids is that they have to have this nitrogen element in a heterocyclic ring, which just means it's a ring structure that has more than one element in it. And they have to be derived from amino acids. So alkaloids play multiple roles when thinking about insects and insect physiology. So what they can do is they can affect insect enzymes, they can inhibit insect DNA synthesis and repair, and then importantly, they affect the insect nervous system. And this can cause paralysis and other toxic effects, mostly because these compounds interfere with postsynaptic receptors. And a few examples of compounds that do that include nicotine, which comes from tobacco, which I'm showing you right here, Nicotiana attenuata, as well as conline and conicine, which are derived from poison hemlock. And so you might be familiar with poison hemlock because it has long been known as a poisonous plant and was actually used to put Socrates to death for corrupting the youth. So its toxicity is due to this group of piperidine alkaloids. Like I said, and conine and conicine are members of that group. And it was really the conicine in Socrates' cocktail that caused paralysis and eventually his death. And so the way that it paralyzed Socrates is the same way that it paralyzed insects, which is basically targeting those postsynaptic receptors. And what happens is it causes muscular paralysis of the respiratory muscles. And because of that, then, you can't really breathe and you don't have oxygen, and that oxygen then can't get to your heart and brain. And so it results in death. So not great. I don't recommend drinking that. Okay. So the next group are cyanogenic glycosides, and these are found in 10% of all plants. And they are also derived from amino acids, but only 6 amino acids. So basically with these compounds, you have a sugar. So here would be a sugar that is bound to another functional group. And the reason the sugar is so important is that having that bound to that functional group keeps them stable. And in their stable form, they are usually stored in vacuoles. And then when the leaf is injured or chewed, those vacuoles burst, and they are then floating around the cytoplasm, and those sugars can be cleaved, and hydrogen cyanide is produced. And that's what really makes them toxic. And I'm going to go into that a little bit more in the next slide. But in this slide, what I want to talk about are peach pits and bitter almonds, both of which contain cyanogenic glycosides, amygdalin, which I'm showing you here, and also prunasin. And these can have fatal results for humans. So of course, it depends on the concentration, which can vary, and also the size of the person ingesting them. But it's usually about 2 peach pits that are pulverized, or a cup of 2 or 2 of bitter almond that will cause mortality in humans. So again, don't eat that. Okay, so how is the sugar moiety cleaved from these compounds to make them toxic? When do they become toxic? Well, when a cell is wounded by an insect feeding, the stable cyanogenic glycosides that are usually stored in the vacuole, which you're seeing here in a scanning electron microscope image of a plant cell, when it's fed upon, this vacuole is going to break open, and all the cyanogenic glycosides stored in them will come in contact with hydrolyzing enzymes like beta-glucosidase that are present in the cytoplasm, and that will break them down. So what happens is the sugars are released through hydrolysis, And then a cyanohydrin is formed, and that decomposes spontaneously to prussic acid or hydrogen cyanide, which I'm highlighting here, and a carbonyl compound. So the hydrogen cyanide is the real kicker, and that affects the mitochondrial respiratory pathway, and that's what results in oxygen starvation and then eventually death. So an interesting example of this in humans is with bitter cassava root, which is the major source of carbohydrates in South America, and actually the third largest source of carbs in the tropics after rice and maize. But it also contains these cyanogenic glycosides. So the traditional way to prepare bitter cassava root for consumption is that you first need to peel it, then you have to grate the root, and then there's this prolonged soaking period where the gratings have to sit in water, and that allows leaching and fermentation to take place. And then after that, they have to be thoroughly cooked, and the cooking is what's really releasing this volatile hydrogen cyanide gas. And then you can eat it. But basically, if they're not properly prepared and cooked, a single yucca root sold in your grocery store could actually kill you. Okay, so let's move on to glucosinolates, which are mustard oils. So these compounds not only contain nitrogen, but also contain sulfur. So you can see that up here in this molecule where you have a sulfur and a nitrogen in that compound. And they also are derived from glucose and an amino acid. So here you also see that glucose moiety once again. The glucosinolates are natural components of lots of different pungent plants like mustard and cabbage and horseradish. And that pungency is due to the mustard oils that are produced from glucosinolates when the plant material is chewed or cut or damaged. So maybe you've already caught on, but these compounds are biosynthetically related to the cyanogenic glycosides that we just talked about, in that they're both spatially separated from the hydrolyzing enzyme and become active once they are in contact with that enzyme, and that glucose moiety is released. So in this case, the hydrolytic enzyme, though, is myrosinase, and glucosinolates come in contact with that upon tissue damage, once again, from herbivory. And then the unstable aglycones, which is really the resulting molecule when the sugar is cleaved, those are released. And then that can result in lots of different active compounds, including different nitriles, isothiocyanates, thiocyanates, etc. So it's really these aglycones, or compounds without the sugar or the glucose, which got cleaved back up here, that tend to react spontaneously with compounds containing unshared pairs of electrons, mainly molecules like proteins and amino acids. And when these glucosinolates, or the active part of the glucosinolate, reacts with those, it makes them inactive. So you can obviously think of a lot of different ways where that would be detrimental to an insect. And then finally, I want to talk about non-protein amino acids. So they are widely distributed across plant taxa, but are incredibly characteristic of legumes in the Fabaceae family, as well as grasses in the Poaceae family. So they are the simplest nitrogen-containing compound that have known toxic effects, I should say, on insects. So if you'll recall, there are about 20 essential amino acids, but there are over 400 amino acid mimics that can be toxic. And I'm going to give you one example here with cannabinine and arginine. So cannabinine from jack bean is actually a structural analog to arginine, which is an essential and protein building amino acid. So cannabinine is the mimic and L-arginine is the essential amino acid. And you can see just how similar they are, right? But, even though they're similar, they're not the same, and they have different properties. And so the RNAs, they can't distinguish during protein synthesis, which is arginine and which is cannabinine. And so they can incorporate cannabinine during protein synthesis. And again, structure dictates function. So by incorporating the wrong amino acid, that's going to alter the 3D structure of the protein, and that's going to lead to a change in the function of the proteins. So those changes in proteins can cause kind of anti-metabolites, right? They serve as anti-metabolites because they can provide non-functional proteins. Sometimes the proteins that are made actually interfere with essential enzymes. So instead of making the protein or enzyme needed, let's say for development, for maybe the hardening of a cuticle, you don't make that protein and the cuticle doesn't harden, right? They can also serve as neurotransmitter mimics. So they can mimic neurotransmitters like dopamine and norepinephrine. And so all of those obviously can be detrimental to herbivores in some way. I guess on a positive spin is that there's been some research to suggest that insects can actually break down non-protein amino acids and use the nitrogen within those compounds. So there might be a trade-off, but this has really yet to be shown as a major source of nitrogen for insects. Okay, so there you have it. So those are the nitrogen containing compounds and some examples. And in my next video, I will talk a little bit more about the next group of compounds, which are terpenes. So thanks everyone, and talk to you soon.